Okay, so today we are going to talk about molecular dynamic simulations in detail. Okay, so first let me introduce you what is exactly molecular dynamic simulations. Thereafter, let's dig into more details like how we usually do this and all. Okay, so the thing is that I hope you all know the difference between a photo and a video, right? So what is the difference between a photo and video? It's like we are getting only a single snap here. So based on this single snap, we construct the memories, or here in this case of docking, so we analyze the interactions and we'll look into how the things are happening between the ligand and the protein, or protein and the peptide, or one protein and other protein. But in the case of video, we can see, like, suppose in the here in this case we'll have, this is the protein and this is the ligand, and here it is sitting like this. But in the case of this molecular dynamic simulations, so this is actually what is called a docking, and which we usually compare with the photo. Okay, so in the case of molecular dynamic simulations, it's like a video. Okay, ma'am. So what happens here we have a protein. Okay, so what is happening here, suppose here we have the ligand. So this ligand is slowly moves to the active site cavity, and finally it fills the active site cavity. Okay, so now we will end here. We get only the set of interaction between ligand and the protein. Okay, but here in this case video means it has certain time, right? So like let's assume, like, in a general sense, suppose we have a video of 30 seconds. So within this 30 seconds time frame, how this ligand is moving. Okay, so how the ligand is moving, not just the ligand, even the protein also moves. Okay, how the protein is moving, and during the course of this movement, is there any change in interactions? Okay, and if you remember, in the case of docking, we were talking about binding affinity, right? So here we are talking about binding affinity. So even in the case of molecular dynamic simulations, we will be talking about binding affinity. Okay, with reference to time also. Okay, in every second, at every second, how the energy is changing, how the interaction energy between the protein and ligand is changing. So all those things we are going to analyze in the molecular dynamic simulations. Okay, ma'am. So like there are many interesting observations uh, which has been published. So we'll look at each and everything in detail now. Okay, I hope with this little info we can move forward. So this is like, when you talk about docking, it is this an age old strategy. It's like this is very old now for understanding the molecular interactions. Even though it is old, but it is very significant, and still even today we can publish papers just by doing docking, and with a good justification of why you are doing the docking. Okay, so now you can see here two different pictures. Okay, so here you can see this is the entire protein. This active site cavity is very deep. Okay, active site cavity is very deep. So here the both the proteins, let's you can see the almost both the proteins are same. Here also the cavity is here. Okay, so this is the cavity, it is deep inside. Here also it is deep inside. Now you can see, can you guess? So this, this is the ligand is very big, and it cannot enter this space. Okay, even the same thing holds good with this also, but it has entered inside the cavity. So how it is possible? Yes, ma. Like using docking, we can't understand all these things. Okay. So if you give a protein, it will go and dock here. So this is what happens. But so this is more or less like a docking. Okay. But if you do simulations, the this same molecule, it will go inside and it will sit in the active site cavity. So let's assume this is molecular dynamic simulations. Okay. So how it is possible? How the ligand is traveling inside the protein? Okay. So how it is possible? Can you guess? Yes. So what
So
happens? Yes, Ramesh.
is it due to the intermolecular
Intermolecule is
spacing in between?
spacing. Okay. If you remember in this previous slide, I was mentioning the protein is moving. Okay. So what happens here? The protein once as the ligand keeps entering into this space. So because of the what we are doing MD simulations. So the protein also keeps on moving. So the protein will open up and it will allow the ligand to enter inside. Okay, that is the reason here the ligand is going inside the protein. Okay, is it clear? In this case, the protein structure is not going to change. Okay, the protein stays as it is, or even if it has some changes, the changes will be very minimal. But in the case of molecular dynamic simulations, the ligand and the protein, they keep on moving continuously. So because of this continuous movement and interactions, so the protein will open up and it will go inside the cavity. Okay. So I will show a few examples to understand these things in detail. Like if you see here, so why we are actually doing simulations? This is because mimic the natural process. Okay. So what happens inside in our body, exactly that we want to do. Okay. So to understand those things in detail, we are doing molecular dynamics simulations. Okay. So if you see this one, so just observe this video very carefully. The protein changes its conformation with time. Okay, can you see here this upper region? It is coming down. Okay, and this one loop it is going towards this cavity. So you can see here there is a cavity, and this is coming down, and this loop is also going up. Okay, yes, are you able to observe? So this is how actually the proteins keep moving inside our body. Suppose actually here I have removed the ligand molecule. Okay, now. So here actually it has one ligand molecule. Oh my God, now this one. So here we have one ligand molecule here. So to bind to that ligand strongly, this region is coming downside and this loop is going this way. Okay. Now. Yes, I hope you are able to get. So even that is not just with the protein, even the nucleic acids also, they keep on moving. Okay, so you can see this is one interaction from the ribosome translation process actually. And here you can see this is the loop. Okay, ma. Are you able to see my pen? Sure. Now this does. Okay, ma. So can you see here? Here we have this loop. Okay, and there is another one, this is moving this way. Okay, this is the RNA molecule actually. Okay, and now you just observe, so this region it was actually like this. Okay, So it is changing its position this way. It is like opening up. So it is allowing the RNA to move inside actually. Okay, ma. So because of the different view, you couldn't visualize it in better way, but you just try to understand this thing. Where is the play button? Okay, so you can see this one. It was like a straight line and it was moving upside. And now this RNA will slide into the this thing. Okay, ma. Inside the ribosome. Okay. So why we are looking all these movements? Okay, even if you see all these movements, how you are going to uh, extract information or knowledge from these movements? That is very very important. Okay, ma'am. So let's discuss all those aspects. Before going into those aspects, let me introduce you some basic terminology. Okay, so here there are some basic calculations. So once you are done with MD simulations, like once you are done with molecular docking, what we are doing? Yes, ma. Once you are done with molecular docking process, what we are doing? We are looking at the interactions between the protein and ligand, or the protein and the protein, or protein or peptide. But here we are not going to look at the interactions. We are going to look something more. Okay, we are going to look something more in detail. Okay, let's discuss what all those things. Okay, first we are going to look at the thing called root mean square deviation. Okay, ma, suppose if this is the conformation of the protein, so how this protein is changing its conformation with respect to time. Okay, ma, suppose let's take an atom. Okay, so what is the position of the atom? Okay, what is the position of the atom at time t is equal to 0, and what is the position of the atom at time t is equal to some n? Okay, so this is all what we are going to look into these things. So how the position is changing? Okay, because suppose if you are simulating this thing with the ligand, so is there any sudden change? So all such aspects we are going to look in more detail. Yes, Ramesh?
Sir, like we are using t is equal to 0 to some indefinite time, sir. So
No,
this
no,
would
it is
require a
not
bit of
indefinite time. Okay,
yes, sir. To
this is
some
not indefinite time. Huh?
with
So
some fixed time.
yeah, it's some fixed time. Even it is not in seconds. Okay,
Okay.
so that I will get back to you in next step. Okay,
Yeah,
so
yes,
first
sir,
let
sure,
me
sir.
give you a glance, then I will tell you. Okay, you usually it's like we are not going to measure here it in seconds. Okay, so we are going to measure in terms of 10 to the power of minus 9 to the maximum of 10 to the power of minus 15 seconds. Okay, ma. So 1 second you are going to divide into these many times, 10 to the power of minus 15. So this is the maximum time, that is femtoseconds, and this is nanoseconds, and this is femtoseconds. So to this extent we are going to divide the time, and we are going to study the interactions. Okay, so here if we look at this thing RMSD, so suppose you can see here, so this is RMSD of one protein interaction. Okay, one protein is interacting with the another protein. Okay, I have given a color codes also here. So this black one is the protein complex, and blue one is the protein 1. So let's assume this is P1 and this is P2. So this blue one is associated P1 and this is P2. Okay, so now you can see here this black one is the complex. So you can see here what is the major influencing factor in this out of this P1 and P2. So P1 is the one which is majorly influencing the movement of the complex. Okay, ma. So but in this case you can see this is more or less it is having a stable, it is like a straight line, but here you can see this has some changes, and these changes are they are reflected in the complex also. Okay, So here what we are not going to look at this. Mostly we'll look at things as such. It should be a straight line. Whenever there is a complex, it should be a straight line. Okay, this is more or less like a straight line, but in some cases we will see a graph as such. Okay, this way. So can you see what is happening here? At this particular point of time, there is a sudden change. So there is a sudden change at this particular point of time, and what are the changes happening, that we are going to observe in more detail. And you can see after changing, I am saying drawing it more or less as a straight line. So it means after changing the conformation also the protein is stable. So the complex is stable. So we'll look at what is exactly happening at this point. Okay, so we'll look at the video part and we'll see what exactly happening. Okay, why this sudden shift is here. Okay, so we'll look in the movement of the uh, amino acids. Okay, so which amino acid or which what is happening, suppose if you are talking about protein ligand interactions, so what is happening to the protein or ligand at this particular point of time. Okay, so all such things we are going to look at the things. Suppose let's assume like till this point of time, so we have a protein like this. Okay, so the ligand is staying here, and all of a sudden at this particular point of time the protein has opened up and the ligand has went in. Okay, so because of this sudden change in conformation of the protein, so we can observe this difference in the RMSD. Okay, I hope you are able to understand the point. So how to interpret this graph in the uh, terms of interaction or in terms of change in structure of the protein or protein ligand complex, that is what we have to look into the details. So just by looking at the graph, the graph looks is like a straight line, it is stable. Okay, this is like a layman language. So even if you, there are many such papers got published, but that is not the right way to talk about the molecular dynamic simulations. Suppose whenever there is a huge difference, so you need to look at the MD simulation. We call it as not movies, we use the word called trajectory. Okay ma, so here we use the word called trajectory. So we need to watch this trajectory of the simulation complex, and we need to analyze exactly what is happening there. Okay, so that is what we have to report in our uh, observation. Okay, so now we have another thing called root mean square fluctuation. So sir, this is only about the protein, this is about whole protein or whole protein ligand or whole protein complex, but now you want to add some more data. So then in that case we can get individual movement data for each amino acid. Okay, so we can get individual movement data, how one amino acid is changing its conformation with respect to time. So that we call it as root mean square fluctuations. You can see here I have shown a graph for 0 to some number. Here it's around 260 amino acids. Okay, so you can see here this is like more or less like a straight line. Okay, so these amino acids are like a straight line. You can see this region, so this region is having a very high fluctuations. Okay, so till here, till this 175, it is most of the amino acids are stable, and here one amino acid is having a good peak, this one. So we need to make a note of these amino acid numbers. Okay, and while you are looking at the protein ligand complex, when you are looking at the protein ligand complex or protein protein complex, then at that point of time you need to focus on these amino acids, how this amino acid conformation is changing with time. So that you have to focus, and you need to draw your observations or conclusions. Okay, so you can see this region, it is more flexible. So this may be like out of the, suppose the protein is something like this, and only this region is interacting with the protein. Okay, so this is one protein and this is protein 2. So you can see only this region is interacting with the protein. So this region is free. So this will be more flexible. Okay, this region will be more flexible. So that is what we can see here. Okay, so this region, this second protein has two different domains or two different regions. One region is interacting with the one protein, another region is free. So it is keep on moving. So this is more flexible as this is having interactions with the another protein. So this is like somewhat stable. Only the functionally important amino acids falling in this region, they are moving. Okay, ma. Okay, so then this is about the root mean square fluctuations. So it's like there is another term called radius of gyration. Okay, ma. So what is mean by radius of gyration? So when you are talking about rotation of the protein, okay, ma. So when you are talking about rotation of the protein with respect to x-axis or y-axis, okay, how the structure of the protein is changing with respect to x-axis, how the protein structure is changing with respect to y-axis. So that is called radius of gyration. Okay, ma. So you can see if you look at the our previous slide, this one. So there are major changes here in this, not this one. So you can see here with respect to the y-axis there is a huge difference of this loop. Actually it is completely going away. Okay, ma. So this is called, when you calculate this, it means there is a movement or change in configuration with respect to axis, x-axis or y-axis. That is called radius of gyration. Okay, so usually most of the proteins they have a stable radius of gyration only because they won't flip the entire structure. Okay, and another most important parameter what we have discussed in docking is binding affinity or binding energy. So here also we calculate binding energy, but we calculate for every frame. Okay, ma. So it's like every nanosecond, or you can calculate more in detail also. So you can see here this is the energy change with respect to time from 1 to 100 nanoseconds. Okay, so here you can see there is a huge fluctuation in the Okay, the recording is in progress actually. So, so we are talking about this free energy estimation. Like here we will be calculating the change in energy with respect to time, and overall we'll calculate, take an average of all these things that we will be quoting in our paper. So sometimes it is not mandatory that you need to consider from the starting to the ending. You can take a part of the uh, time also, and you need to justify why you are not taking only that particular time frame. Okay, so if you have a justification based on the concept or the things you are studying in, then it is fine. If you are not providing any justification, then that would be a problem actually. Okay, so next we have H bond analysis. So we are also, as you already know, like in case of molecular docking, so we are preferably looking at the hydrogen bond interactions. Okay, but some ligands they don't have any hydrogen bond interactions also. But here in this case in MD simulations, we will be looking at the hydrogen bond interactions with respect to time. Okay, ma. So with time, how the hydrogen bond interactions are changing. So you can see here there is an average of 12 hydrogen bonds, and we have a very few minimal hydrogen bonds of around 8, and maximum is 16. Like, okay, so how these things are happening and what is happening exactly at that particular time point, so we can look into more details. Okay, so but here in MD simulations we don't focus more on these hydrogen bond interactions. Okay, so we will be focusing on these graphs and these graphs. So we want to look at how the protein or ligand, how the interactions are changing, how the amino acids are moving with respect to time. Okay, so that is what we are mostly interested in. Okay, suppose there are some case studies as such. So we have a ligand. Okay, so this will slowly go and reach this area. So this ligand will slowly goes and reaches this area. Okay, but and now the question comes, till what time it will stay here. Okay, so suppose there is another ligand, let's assume it is B. Okay, so it has went here and it got struck here. Okay, let's assume there is another ligand C. It went inside and it also came outside. Okay, it went in and it also came out. So all such fantastic observations we can make using molecular dynamic simulations. Okay, and there are something else like clustering analysis and principal component analysis. So you know, in Haddock we have heard the word called cluster. Okay, ma. So the similar poses, all of them will be grouped, that is called clustering, and based on the clustering we'll draw some XY graph. Okay, so that is called principal component analysis. Okay, ma. So this will be a bit higher for you guys right now. So like there are some advanced things also. So this, like we have pocket volume measurer. This comparative to when you talk about principal component analysis, so we are identifying the key residues or key aspects there. But here in this pocket volume measurer, this is something advanced level. Usually you don't see it in most of the research publications. So this is like how the active site cavity of the protein is changing with respect to time. So let's assume, so this is the active site cavity. So how the size of the cavity is changing with respect to time. So that is called pocket volume measuring. Okay, ma'am. So you can see there are some different set of proteins. Some proteins, so you can see the black one, it is having more or less like a straight line. And you can see this green one, it is having a decreasing trend. The pocket volume is reducing with time. Okay, so you can see some protein, this one which is in light brown in color. So this you can see the protein's volume is increasing with time. Okay, ma. So this is following a trend this way. Okay, So different proteins behave differently with time. So all of them are binding to the same protein, but they behave differently. And why these differences are happening, that is what we are actually interested to look in more detail. Okay, so that forms a very good publication. Simply you put most of the research papers, what they do, they put all these graphs and they will tell maximum number of bonds, minimum number of bonds, average binding energy. So this residue is having more fluctuations. That's it. So they are done with their story. So that is not, that is the observation. Okay, or that is the output of the this simulations. But based on this observation or output, what conclusions you are drawing? It means to draw the conclusions, as I mentioned, you have to look at the trajectory. Okay, what is trajectory here? It is like a movie. Okay, so we have to look at the trajectory and we need to correlate these changes with in the trajectory. Okay, what is happening exactly here? Then that is actually what you have to report, okay, for a scientific publication or in your thesis. Then that forms the good thing. So there are some additional thing called residue interaction network. So how the residues are interacting with respect to time. Okay, ma. So not just with time. This, like, we have taken average structure and then we have looked at the interactions. You can see here these are the active site residues. Okay, more or less their names will be same. Okay, ma. You can see here all of them have adenine. Okay, adenine and adenine, and you can see here we have tyrosine, tyrosine and tyrosine. So when this tyrosine is mutated to valine or glutamine, we don't see any interactions. Okay, this is the one of the key observation. And you can see this set of residues: tryptophan, arginine, glutamine, tryptophan, lysine, glutamine, tryptophan, arginine, glutamine, tryptophan, arginine, glutamine. So what is the difference between arginine and lysine? So arginine and lysine, both of them are like amino acids which contain the amine group. Okay, so they are having same properties. So that is the reason these interactions we can see in both uh, these all the five proteins. Okay, so that is how we observe the similarities and differences in the protein and we characterize them. Okay, so is this all enough? No, this is all, it is not enough. Okay, that is what I am saying actually. So we have this two set of proteins. So what is happening exactly at this particular location? So we have to zoom in and see. Okay, ma'am. So you can see here there is one thing, this is in blue color and this is in red color. Okay, so this is at time t is equal to 0 and this is the red one is at time t is equal to 90 nanoseconds. And you can see here this red colored one, here we have an amino acid called serine. So in the blue one it is exactly, it is at this location, but at time t equal to 90 seconds this has come down and it is facing towards the ATP. It means this is trying to interact with the ATP. So this kind of things you need to observe in the trajectory, and that you have to create some beautiful pictures and report in your publications or the thesis. Okay, and next another thing is that here at this location we have arginine residues, and you can see this arginine residues here, it is like three arginine residues. One arginine is like this blue one. Okay, it is facing downside, but slowly later this also moved up. You can see this is moving like a straight line. It was down and it moved up. So it means it is also going away from the interactions. Okay, so that is how, these are the things actually we need to observe and report in our publications. So this is about an overview of molecular dynamic simulations. Okay, like what is MD simulation, what type of calculations we do, and what type of peaks we have to generate or how to interpret the result. Okay, so this is in brief about the MD simulations. So if you have any doubts in this conceptual part, you can unmute and ask. So after this, let's discuss in detail about the procedure and all other aspects. Okay, now. Yes, now the session is open for doubt clarification. You can unmute and ask.
So in that previous graph that you showed, like there was a sharp
Yes, ma. So
inclination.
is that mean no doubts?
No, sir, there was one graph with the black line which sharply inclined.
This one.
No, sir, before that.
This one, no? No, these are the only graphs, right? I was showing.
Which had a steep
So these are
inclination
the
and then
graphs I have shown.
became
This.
stationary.
So which graphs you are taking? This one? No. You can see which graph you can tell. This one. Or this one, I think. This is the black line. Like we have an inclination in the initially. Initially we have inclination, then it has becoming
Yes, sir. So
like
for these kind
you
of
can see got
graphs,
decreased and
do you
then
do the area
again
under
it
the
got
curve
stable.
method for
This one?
finding the?
No, no, we are not talking about area under curve. Okay, we are talking about okay, this is surface area graph. Okay, so no, here we
Okay,
are
sir.
talking about how the volume of the pocket is changing with respect to time. Okay, this is not about the area under curve. Okay, any other doubts? Okay, so means if you guys don't have any doubts, then one second. Then let's move on to the methodology part, like how we do this MD simulations and all. ఇందాక వచ్చిన లైన్ సరే ఓకే okay ma, so let's have a break here for 10 minutes and you guys join back by 8. Then at that point of time let's discuss the methodology part. Okay, a brief overview of the methodology part. Okay, so because most of the things it doesn't support with our laptops, and we can't do these things online also. Okay, we need the software to be installed and we need a graphic card. Okay, so at least of 10 GB graphic card, so then only we can perform the MD simulations. Okay, so I will introduce you to the method